Hello, today we're going to talk about treating cancer using gene editing. So, in this presentation today, we're going to understand the basics of cancer and traditional treatment methods, learn about gene editing technologies focusing on the CRISPR-Cas9 method, as well as explore how gene editing is being applied to cancer treatment. So, what is cancer? Cancer is a group of diseases characterized by excessive cell growth and spread of abnormal cells. Now, these cells can invade nearby tissues and organs and spread to other parts of the body, which this process is called metastasis. Now, how metastasis actually happens is that when a cancerous cell leaves the original primary tumor, travels around the body using the lymph system or the blood system, and stays in another organ and creates and forms a new tumor and engages and invades another organ as well. That is called metastasis, and the cancer has become even worse now. As in for different types of cancer, we're going to talk about it in the next slide. But for cause of cancer, there is three different or even more factors that can cause cancer. We're going to talk about the three most common ways. The first one is the physical factor of cause of cancer. It's usually characterized by radiations such as UV rays, ultraviolet rays, gamma rays, X-rays, microwaves. The other one is biological factors such as a weakened immune system or a viral infection, and a viral infection such as HPV, HIV, hepatitis B, hepatitis C, and also chemical factors such as being exposed to tobacco smoke or being exposed to dirty weather full of pollutants. These are all different factors that can cause cancer. Now, these are all different types of cancer. In human, we have the throat cancer, the esophagus cancer, lung cancer, stomach cancer, bowel, bladder, skin cancer. We have prostate and testicular cancer only happening to men, and also breast cancer, ovarian and uterus cancer only happening to women. Now we're going to compare a normal healthy cell compared to a cancerous cell. Now in normal cells, the shape of the nuclei is pretty small and is also defined. It's uniformly shaped. And the cytoplasm is quite large in normal cells. However, in cancerous cell, the nuclei is very big. But contrary, the cytoplasm is very, very small. Secondly, in normal healthy cells, this size of the cell, the shape of the cell is, pretty certain. And also the arrangement of the cells are pretty organized. We know what they look like, for example, in this. However, in cancerous cells, the shape of the cell and the size of the shell varies. It's not really organized. And also the arrangement of the cells are unorganized. As we can see here, the size and the shape of the cell are different for each cell, and they're put next to each other in an unorganized fashion. Third difference is that normal healthy cells have intact differentiated cell structures such as a nucleolus, nucleus, ERs, Golgi apparatus, and many other ones. However, in cancerous cells, the cell structures have been lost or don't exist due to mutation. Last but not least, in normal cells, cell division is at a normal level and only occurs when the cells receive a signal to do so. However, in cancer cells, they don't receive any signal for cell division. And because they don't receive any signal, they just excessively divide over and over again in a large number of dividing cells, as it says here. There is no control over how much they divide. They just keep dividing because nothing stops them. Now we're going to talk about traditional cancer treatments. The first one is a surgical operation. It's the removal of the tumor. The benefit of this treatment is that it entirely removes the tumor at one go. It removes a large quantity of cells, cancer cells, in one go. But the downfall with that is that it doesn't remove all the cancer cells. So at the end, some cancer cells still remain in the patient's body, which increases the chances of another tumor being formed. Second one is radiation therapy. It uses a high dosage of radiation to shrink the tumor and kill cancer cells. The downfall with this method is that the radiation can also affect and cause long-term damage to the nearby tissues and cells. Some of the side effects are skin irritation, bruises and bumps, dryness in the body, and also, as I said, it causes long-term and permanent damage to the nearby organs. The third and probably the most common way is a chemotherapy. It uses drugs to kill cancer cells. The downfall with that is that the drug that is being used goes through the body using the bloodstream, so that all the cells, the entire body is affected by the drug. That's why people who undergo chemotherapy has really weak immune system, their hair falls off, and that's all because of the drugs in their bloodstream. Last but not least is nuclear therapy. It uses radioactive substances to shrink and kill cancer cells, pretty similar to radiation therapy. The downfall of it is that it can affect and cause long-term damage to the nearby tissues and cells. Now we're going to talk about gene editing. Now, what is gene editing? Gene editing is a set of technologies that allows scientists to change an organism's DNA. Now, gene editing has changed the field of genetics by providing tools that allow for precise modifications to the DNA. Now, among these tools, CRISPR-Cas9, which we talked about before, has gained a lot of attention due to its simplicity and efficiency. Now, what is the purpose of gene editing? The purpose of it is that it gives scientists the ability to make precise alterations to the genetic code, potentially correcting mutations that cause diseases such as cancer. Now, there's different types of gene editing technologies. One of them is CRISPR-Cas9. It's a powerful tool that allows for precise, direct changes to the genome, to the DNA. It uses a guide RNA to target specific DNA sequences and an enzyme called Cas9 enzyme to cut the DNA at the target location. Then we have TALEN, stands for transcription activator-like effector nucleases. They're engineered proteins designed to target specific DNA sequences for cutting, only for cutting. And then we have ZFN, zinc finger nucleases. They're still engineered proteins. Another type of engineered protein, that can be customized to target specific genes for only editing. Now, the advantage of gene editing is that it has the ability to make precise changes to the genetic code. It has a big potential for treating genetic diseases such as cancer, right? It is pretty precise, as we talked about, right? It minimizes off-target effects, contrary to radioactive and radiation therapies, and also chemotherapy, which they affect the entire body. Gene editing, it minimizes off-target effects. It only affects a specific gene. It is efficient because it can make precise changes pretty quickly compared to other traditional methods. And it's also versatile. It can be used to insert, delete, or repair genes in a variety of different organisms, like in plants, in humans, in other animals, or even in bacteria. Now we're going to talk about CRISPR-Cas9 method in detail. Now, it uses a guide RNA, gRNA. It's a synthetic RNA molecule that matches the sequence of the target DNA. It directs the Cas9 enzyme to the exact location in the genome where the genetic modification needs to be made. As we said, it uses a Cas9 enzyme. The enzyme Cas9 is a protein that acts as a molecular scissor to cut the DNA at the specified location. Now, once the DNA is cut, the cell's natural repair mechanisms can be used to introduce changes to the DNA, which we'll talk about later. This method has a lot of advantages. One of them is that it is pretty predictable. The reason it's predictable because it is a DNA to RNA interaction compared to other methods where it's a DNA to protein interaction, which makes it less predictable, which makes them less predictable. This method is, we can predict what happens during the modification and even after the modification using CRISPR method. It's also pretty affordable and less time consuming than other methods. Now we're going to talk about the step-by-step review of what happens in CRISPR method. First of all, we have to design our guide RNA to match the target DNA. Secondly, our guide RNA then goes on to bind to the target DNA. Thirdly, the Cas9 enzyme is guided to the location and makes a double-stranded break in the DNA, which looks like this here. And then four, the last step, technically, the cell's natural repair processes are harnessed to introduce desired changes. Now, the natural repair system uses two methods to do so. The first one is called a non-homologous end joining, which stands for NHEJ, the body's default repair mechanism, often resulting in insertions or deletions that can disrupt the gene. As we can see here, right, it deletes a gene and the gene is disrupted. It just connects to two ends of the gene together, and it's usually a deletion. The second method is called a homologous recombination, or HR method. It's a more precise repair mechanism that uses a provided template to introduce specific changes, which scientists can supply during the editing process, which looks like this. Using an artificial template provided by the scientists, a new sequence is added to the gene or is introduced to the gene. It inserts a gene there. Now we're going to compare different technologies of gene editing between ZFN, TALEN, and CRISPR Cas9. Now in the construction of these three methods, ZFN and TALEN, they are pretty similar to one another. They're engineered proteins for every single target. So every single target needs to be engineered in a different way. However, for CRISPR method, it's a 20 nucleotide sequence of guide RNA that will be transported to the specified location. For targeting, ZFN and TALEN have a protein DNA interaction, which makes them less predictable, as I talked about before. But the CRISPR method makes a DNA RNA interaction, which makes it highly predictable. So we can predict what happens during the editing and after the editing. For the delivery, for TALEN and ZFN, two molecules or two ZFN or TALEN need to be around the target sequence. But for CRISPR, a guide RNA complementary to the target sequence with Cas9 protein needs to be there to match the target DNA. And then for multiplexing and feasibility of library reconstruction and transforming for genome wide screens, both TALEN and ZFN are pretty challenging to do. but CRISPR is highly durable. They're pretty feasible. As in talks of affordability, ZFN and Talon are resource intensive, time consuming, and expensive, but CRISPR is pretty affordable. Now, how is gene editing used in cancer treatment? First of all, it's used for targeting cancer cells. How is it used? So, it can be used to knock out a gene in a cell. Now, it involves disrupting specific genes that are essential for cancer cell survival or proliferation. For example, targeting oncogenes, genes that drive cancer, for example, to disable their function. That's one way that we can use gene editing to knock out a gene to target cancer cells. The other way is gene correction, is correcting mutations in tumor suppressor genes that normally help prevent cancer. For example, a gene that was before used to be a gene to avoid and prevent cancer now has been mutated and is helping cancer cell to divide excessively. We can correct it to make it to go back to what it was before to avoid cancer cells using gene editing. Another example is restoring the function of mutated p53 gene. It's a key tumor suppressor. We can restore the function, correct it so it can go back to what it was before. Second usage of gene editing is enhancing immune system. It's used in CAR T cell therapy. Now, T cells are a type of white blood cells. Now, engineering a patient's T cells to express antigen receptors that target cancer cells. Now, T cells are modified in the lab to recognize and attack cancer cells more effectively. Or another way is the TIL, tumor infiltrating lymphocyte therapy. Pretty similar to CAR cell therapy, genetically modifying TILs to, we can enhance their ability to recognize and kill cancer cells. Pretty easy. The third one is overcoming drug resistance. It's targeting resistant mechanisms in body, editing genes involved in drug resistance pathways to improve the effectiveness of existing treatments. Now, an example of it is that modifying genes that contribute to resistance against chemotherapy drugs. If the body creates resistance against chemotherapy drugs, now that drug is not as effective anymore against cancer cells, and cancers just keep on dividing and dividing till they overcome the person. So by adding a gene that contributes to drug resistance, if we don't allow that gene or allow the body to become resistant against the drug can easily do its work and kill all the cancer cells. Well, that was it for today. Thank you very much for listening. I hope you enjoyed this presentation today. Thank you.